Hello everyone, welcome to my presentation. My name is Snehal Dhananjay Shinde and my topic is pharmaceutical and medical perspectives of melanin pigment from streptomyces species. We done the project under guidance of Professor Vaishali ATA, Department of Microbiology, KTHM College, Nasik. So let's move on the project. So first is introduction. So the streptomyces is a group of organisms widespread in nature and play a significant role in the future of biotechnology. Organism is synthesized and extracts dark pigment, that is melanin or melanoid. It is an irregular dark brown polymer that is produced by the various microorganisms by the fermentative oxidation and have a radioactive, protective and antioxidant properties that can effectively protect the living organism from the UV radiations. The melanin formation is a key feature for the classification of streptomyces species. It is used in the medicine, pharmacology, cosmetics preparation, antioxidants, reducing a risk for the chronic disease like cancer, heart, and many more. So next is an objective to study the antioxidant activity, to explore antimicrobial activity against human and plant pathogen, to decolorize the azodie, heavy metal resistant activity, and last that is copper and silver nanoparticle formation, which can be further used in the treatment of cancer. The given flow chart showing the biosynthesis of the melanin. It is a derivative of a tyrosine which is converted to the dopa, then further dopaquinone, then to the dopachrome, and then auto-oxidized to form 5,6-dihydroxyindole, which is brown form of the melanin, and 5,6-dihydroxyindole to carboxylic acid, black, that is the black form of the melanin. Now the next step is are showing uh, isolation of the streptomyces species and the production and the extraction of the melanin pigment. For the isolation, rhizosphere soil sample collected from the different areas, then the serial dilution technique was performed and strict on the tyrosine casing agar plate, uh, and as well as the peptone agar plate. Now the incubated for the 4 to 5 days at the 30 degree centigrade. Result shows the powdery white colonies giving a black brown diffusible pigment. And next is the for the production and extraction of the melanin. Pure culture of the isolate was inoculated in the tyrosine casein and the peptone iron broth and incubated at the 30 degrees centigrade for the 5 to 6 days. Now the filters of the both broth adjusted to the pH 7, and then the 0.5 gram of the potassium persulfate uh, was added and allowed to stand for 2 hours at the room temperature. Then the 50 ml of the ethanol was added and again it is allowed to stand for the 3 days at the room temperature. After the 3 days, filtrate was centrifuged and at the 10,000 rpm for the 10 minutes. Now the supernatant was discarded and the palette was collected and air dried. So we are using the dry form of the melanin pigment. Now next is the identification and the characterization. The isolated organism was a circular from and having a 2 mm of the diameter, powdery white colonies showing on the tyrosine casing agar, the staining showing the gram positive rod, non-acid fast, and the spirals for arranging, the diffusible pigment producing, which is the brownish black pigment produced by the organism, and catalyst positive is the organism. Now the melanin formation test showing the color change after adding the L-dopa to the filtrate. So the color changes from the yellowish to brownish black. So next is the some physicochemical parameters influencing on the growth of organism. The parameters are checked by the using basal media. In first So the graphs are showing the different parameters. uh, Among these, the first graph is for the effect of carbon sources, uh, which is very effective at the starch. In the second, the L-cysteine is the effective. In the third effective is the temperature is at the 30 degrees centigrade, and in the fourth graph the maximum pH is the 7. From the above all the procedures, uh, the results we found that the isolate organism was identified and characterized as a streptomyces species as per Burke's manual, volume 4. Starch and the L-cysteine was found effective carbon and nitrogen source respectively, and optimum pH is 7 and optimum temperature is 30 degrees centigrade. The tyrosine casein broth was most suited for the production of the melanin pigment rather than the peptone agar, and the estimated uh, sorry, and the estimated melanin pigment was maybe melanin. Now the applications we have done are the antimicrobial, antioxidant, decoloration, heavy metal resistance, and the extracellular synthesis of the nanoparticles. So the first is antimicrobial activity we done against various pathogens like E. coli, micrococcus, bacillus subtilis. So the zone of the inhibition was observed against these pathogens, and some fungi we use like the Aspergillus flavus and Candida albicans. Then the next property we use the antioxidant activity, which is performed by the DPPH method, and the graph we have concluded that showing the dates against the absorbance. So maximum absorbance was observed. So the melanin pigment inhibited the peroxide formation in linoleic acid for the 6 days of the after the incubation. The third application was performed the dye decolorization. Simply the melanin pigment was added to the dye, azo dye, and absorbance was the measure after every minute. So formula we used to calculate the decolorization, that is percent of the decolorization is equals to the initial absorbance minus final absorbance, which is divided by the initial absorbance multiplied by 100. So the initial OD was 0.98 and the final OD was 0.02. As a result was the 95.95% decolorization of the dye was done by the melanin pigment in a static condition after 5 to 10 minutes. Now the next property, that is the fourth property, is the heavy metal resistance performed by the agar diffusion method, and the concentration of the standard heavy metal solution was 100 ppm. So the heavy metal chelate with melanin and shows the zone of precipitation, which is showing the neutralization of the heavy metals. The plates are showing the zone of the precipitation against the various heavy metals. Then the last property is the property of nanoparticles. For this, copper acetate, copper sulfate, and the silver nitrate is used at the concentration of the 1 gram per liter. 30 minutes of the incubation shows the visual observation, that is the color change in the samples, and these samples were used for the characterization and the applications. The bioreduction was monitored by the sampling and measuring the absorbance spectrum, absorption spectrum by the UV visible spectroscopy. Now the future prospects of the melanin pigment are: the melanin pigment can be used to make an organic photovoltaic cell, which can be used to convert the solar energy to the electrical energy. For the preparation of the bioplastic or biopolymers due to the ability to undergo polymerization in the preparation of UV absorbent optical lenses for the bioremediation and the treatment of the disease like leukoderma, and the future of the nanotechnology in the treating the cancer. Thank you.